Welcome students. Today we are going to discuss about the first chapter of class 9, that is fundamental unit of life. In this chapter we will discuss about the cell, which is the smallest particle of our body that help us to form the body. So we will discuss about it. Cell. Cell is the smallest particle that helps to form the body. We can call that cell is the structural and functional unit of life. That means the total organism's body are made up of few number of the cells. How the cells are arranged and also how they are working their functions. On the basis of that organism's body will work. So we can call that cell is not only structural but also the functional unit of life. The term cell is coined by Robert Hooke. Actually, the meaning of the term, that is cell, it is a small room. So on the basis of this one, Robert Hooke first time invented this particular cell from a cork, a wooden cork. He got this one, and from there he gave the term cell, which means the small room. It looks like the small room. So on the basis of that, he coined the term the cell. Size of a cell. Cells are too small that we cannot see them by our naked eyes. So we need some instrument that will help us to see that object in quite magnifying way. So microscope is the instrument that will help us to magnify the object. So under the microscope we can see the cell. This microscope can be of three types. Either it will be the simple microscope, compound microscope, or it will be the electron microscope. Normally in our laboratory we used to get two type of microscope. First one, that is simple, which will help to see the object in a quite magnifying way, and compound microscope, which will help to see the object in a quite broader way. That means in case of compound microscope, a very small thing that we cannot see by the naked eyes, that also we can see under the microscope, compound microscope. In another case, simple microscope, if the object is very small, we can see them by the naked eyes, but their internal structure we want to see, then only we are using the simple microscope. Cell theory. Cell theory was proposed by two scientists, Sheridan 1838 and Shaw 1839. According to them, all the plant and animals are composed of the cell, and cell is the structural and functional unit of life. And then this particular cell theory was further expanded by Virchow, and he include this one, that all cells are arising from the pre-existing cells. All cells are arising from the pre-existing cells. These are cell theory. Shape and structure of a cell. Shape and structure of a cell may vary. Different kind of shape and structure of a cell it can be. So it's not in a particular way that it will be the hexagonal or it will be the circular or something else. That particular shape and the structure of the cell may vary. Primarily, organisms on the basis of the cell can be of two types. If the organisms are made up of a single cell, we can call that it is unicellular. One of the example of it is amoeba, paramecium, etc. Multicellular means if the organism's body are made up of many number of the cells, then we can call that it is the multicellular organisms. And the examples are we human beings. Important parts of a cell. In case of a cell, we can tell that it has three important parts. First one, that is plasma membrane. Second one, that is nucleus. Third one, that is cytoplasm. Plasma membrane is the outer part of the cell. Nucleus is the dense part of the cell. Cytoplasm is the semi-liquid part. Let's see in the diagram. This is the animal cell. So in case of an animal cell, we can tell that the outer layer is the plasma membrane or the cell membrane. Cell membrane and the plasma membrane are same. Cell membrane or the plasma membrane. But in case of a plant cell, we will see it is the second outermost layer, because in case of animal cell, we will see the outermost layer is the cell wall. The next one, that is nucleus. This dense part of the cell is called the nucleus. This is the nucleus. Next one, cytoplasm. the semi-liquid part which is present in a cell, that is the cytoplasm. The semi-liquid part which is present in the cell is called that it is the cytoplasm. So here we can tell, in case of a cell, three parts are important: cell membrane or plasma membrane, nucleus and cytoplasm. Now we will discuss about the plasma membrane. If we will discuss about the plasma membrane, we can call that it is the outer covering of a cell. The outermost covering of the cell is the cell membrane, especially in case of the animal cell membrane or the plasma membrane. It is selectively permeable. What's the meaning of that? Selectively permeable. Selectively permeable means it will allow certain particles to enter inside the cell and it will reject the rest of the particles which are not important for the cell, and they will not allow them to enter. So that's why we can call that it is selectively permeable. It will select the particles which will enter inside the cell. It helps in diffusion and osmosis. Now it's important to know that what is diffusion and what is osmosis. Let's see in this diagram. Here we can tell that diffusion. Diffusion means the process by which the particles from the higher concentration will move towards the lower concentration. Here in this diagram we can see that all the particles, they are very closely packed. That means their concentration is high, and here particles are loosely packed. That means here the concentration is low. So where the concentration is high, the particles will move from this side to the another side where the concentration is low. But here we will not see any membrane or anything which will allow them to pass. That means it is freely permeable. Freely it can move from one to another. So this is the diffusion, where we will not see any membrane or anything that will need to allow. In another case, that is osmosis. The main difference between the diffusion and osmosis is that here we will get a semi-permeable membrane, and in this diagram we can see it is a pure water. That means concentration of the water molecules is high in this side, and it is a solution. That means concentration of the water molecule is low in this side. So where the concentration of water molecule is high from this side, the particles of water will move towards the another side where the concentration of this water is low. This process is called that it is osmosis, and the semi-permeable membrane will help to pass these particles. What will happen if we put a cell into a sugar solution? Before knowing this answer, we have to know that what are the type of solutions it can be. It can be either the dilute solution or the normal solution. Another one is the concentrated solution. Dilute solution means where the sugar particles are very low than the water particles. So in this solution, we can tell that water concentration is more than the sugar's concentration. In another case, in the normal solution, the concentration of water and the sugar will be quite equivalent. So the concentration of water will be quite moderate. In the third one, here we will see that concentration of sugar is more than the water in this solution. That means the concentration of water is very low in this type of solution. If we are going to keep a cell in this dilute solution, we will see that outside water concentration is more than inside the cell whatever the amount of water is there. So that's why the water will start gaining that's why the cell will start gaining the water and the cell will become swollen. So we can see the cell will start swelling. In case of the normal solution, we can see that the outside water's concentration is quite equivalent with the concentration which is present inside the cell. That means water concentration outside the cell and water concentration inside the cell, these two are equal. So the water particles will not move from one to another. In case of the concentrated solution, we can see that the outside water concentration is quite less than inside the cell, the concentration of water. So the cell will start losing the water, and then all the water particles inside the cell, it will start losing and it will go towards the solution, and the cell will start shrink. So here we can tell that this type of the solution is called hypotonic. In case of hypotonic solution, cell will start swelling. In case of isotonic solution, where the concentration of the outside solution and inside the cell, these two are same, this is isotonic solution. Here the cell will be unchanged. Hypertonic solution, where the concentration of the water particles will be less in outside with respect to the concentration of water inside the cell. This type of solution is called hypertonic solution, and in this type of solution the cell will start shrink. So these are the different type of the solutions, and what will be the result if we are going to keep the cell in these three different type of the solutions. Thank you.